And let's start with the first speaker. We have David Manning from University of Nebraska. Let's go ahead.
All right, hey y'all, I'm David Manning, Associate Professor in Biology here at UNL. So just wanted to say welcome to our institution and hope you're enjoying your time here. I really appreciate the Nebraska Water Center coming over to Omaha. And just wander over from all my colleagues to talk to you today about how we can try to answer the question that we often struggle with when we're monitoring to balance monitoring without, thinking about universal metrics or missing some data without straying too far into trying to measure everything all at once. I also wanted to mention that I also serve as Director of Environmental Science here at UNO. So we're always looking for partnerships and thinking about internships for our students. So just a plug to reach out to me if you have any of those opportunities that might be of interest to our program. So I do also want to mention this is a collaborative project. Some of the collaborators are here in the room, Jess Corman, as well as Paul Ayayi, who's here at UNO, and then two students I want to point out, Rodrigo Meza Gonzalez and Vinci Sunny, who are involved with this project. So thanks to them, as well as a really good team of folks helping out in the field or in the lab who are listed here. I want to thank the people who put on this conference, as well as funding from the Collaboration or Initiative grant a couple years ago, and an ongoing grant from the U.S. Geological Survey 114 program. So I'm a card-carrying ecologist, which means that I think about the world in terms of how ecosystems work, both in terms of their structure and their function. And so this is one of the models conceptually that we think about, in that the makeup of the ecosystem in terms of its parts and components are going to feed back and affect ecosystem processes we measure through time, and then those processes would then feed back to affect the living environment. So this is the general structure that we're thinking about, the general model that we're thinking about for how we can monitor water quality in such a way that thinks about structure and thinks about processes and links the two together. So if we're thinking about water quality as ecosystem structure, a lot of the things that we measure already, I would call ecosystem structure, including water chemistry, building blocks for life, nitrogen, phosphorus, and oxygen. But it's also going to include the living things such as these microbes, and some of my favorites, the macrovertebrates down here, we often use for bioindicators, and as well as basal resources. So what's the food that's available to these critters that live in our creeks and rivers? And then from there, these all feed into what we expect to find in terms of ecosystem function. And these are the processes that are ongoing, they happen through time, and they typically are going to be a rate. So things like nutrient limitation, food web and energy flow, how fast and quickly organic matter breaks down, as well as things like primary production and ecosystem respiration. So a big question that we're trying to answer here is to figure out how is it that different management practices and protection status in kind of a binary framework affect a river's water quality, and then how does that link to ecosystem processes? And so I have two fairly simple predictions here, and they are essentially that if we understand the physical chemical features of a river and they're different, then that should have an effect on ecosystem structure, and that can be measurably different and detectable. And the other prediction is that if there's differences in that structure, we should expect to detect different functional regimes. So how does that process change through time within a given river ecosystem? And then ultimately, what I'm arguing here is that this requires measuring these integrated responses that go from different levels of organization, microbial to ecosystem scale. So the two places that we're testing this out, again, are kind of two ends of the spectrum of what we would imagine in terms of poor water quality, good water quality here in the state. And they are the Niobrara River, which is pictured on, well, you might want to guess which one's which. So Niobrara is pictured on the left, and then the Elkhorn River is on the right. And visually speaking, you can see some differences here, but to my eyes at least, they have similar, visual features to them. They both are in our state here in Nebraska, just for reference. The Niobrara River is in north central Nebraska, around here. And then the Elkhorn River is actually about 30 minutes west of where we are now. You can cross over it if you're driving from the west to Omaha. But you can see that the two watersheds eventually drain into the Platte River for the Elkhorn and then the Missouri River for the Niobrara. Two bigger distinctions about the two rivers are the Niobrara River has been a National Wild and Scenic River, and so has protected status under the National Wild and Scenic Rivers Act since 1991. But they're also somewhat similar in that they have the same general drainage area, so about 17 to 18,000 square kilometers. But they do differ in terms of their discharge, where the Elkhorn is about twice as high as the mean discharge in the Niobrara. The other big difference that you might be aware of is just land use and intensity of management that happens in both watersheds. And just to illustrate that, I pulled some data for the manure application rate in both watersheds. And you can see that it's about 0.2 kilograms per hectare per year in the Niobrara, and then it's over 5 kilograms per hectare per year in the Elkhorn. So stark differences in terms of the intensity of land use and what's happening in the landscape in these two watersheds. So with this in mind, what we attempted to do is go out and investigate what's the ecosystem structure like, is that different, and how does that feed into the ecosystem processes in these two really distinct rivers. And so the structural metrics that I'll talk about today are water chemistry, and specifically total nitrogen, total phosphorus. So we took grab samples of water, brought them back to the lab, processed them. We had 4 stations in both rivers, spread out along several kilometer stretch of the river. And then we also measured microbial communities using our RNA sequencing in the 16S section. And we were also able to gather information on functional group analysis. And we collected these communities from multiple substrates. So we have microbes that live in the water column, microbes that live on rocks, basically, microbes that live in the sand. And so we have multiple substrates represented. In terms of basal resources, what we did was we measured how fast leaf litter broke down in these two rivers. And then we also looked at the invertebrates that live in the river, and we collected their stable isotopes. So we took grab samples, or we had a concerted effort to figure out, okay, what kind of bugs are in the river. We grabbed the bugs, we identified them, and then we sent them off for isotope analysis, specifically 13C and 15N. And then with that data, what we're able to do is use mixing models to estimate their diets and what they're eating. So we're using it as a natural tracer to figure out, are they eating mostly leaves and leaf litter and organic matter, or are they eating algae and other resources? So with all these data then, what we can do is calculate things like, how is energy flowing through the food web, specifically through these invertebrates? How fast is organic matter breaking down? And the USGS is also has a sensor out in both rivers to measure DO, and so we can model what ecosystem metabolism is, and that's gross primary production and ecosystem respiration. Okay, so here's what we found in terms of structure, and the way that I've organized this is I'm going to tell you about the structural metrics first, and then try to link to the ecosystem functions after that. And we'll start with the water chemistry. And what you can see is that, similar to some of the data about the physical chemical features of the rivers, just how they look visually, we can see really stark differences in terms of their water chemistry, specifically for total nitrogen, which is on the left, and then total phosphorus on the right. The Elkhorn River is on the left-hand side for these violin plots for both total nitrogen and total phosphorus. And the Niobrara is down here. So these violin plots basically show what's the distribution of the data, and then there's a box plot in between as well. You can see there's a lot of variability in the Elkhorn, so it can be quite low, but it's also quite high. And then somewhere in the middle is shown by these box plots. And then these data are the median levels of total nitrogen, total phosphorus for both rivers. You can see that much higher in the Elkhorn for both nitrogen and phosphorus. We look at the microbial communities, we also see pretty stark structural differences between the two rivers as well. And this is an ordination plot, and the basic idea here is that if you draw an ellipse around where the samples came from the Niobrara, which is shown in these green ellipses here, and then where they came from the Elkhorn River, which is shown with the more burgundy colored ellipses here, there's some differences that we see starting to fall out in terms of the microbial communities. Specifically, we see that in the Elkhorn River, all of these columns here, or rows, I suppose, that are in blue are the groups of microbes that were more abundant in the Elkhorn, and then the ones that were in red are more abundant in the Niobrara. And just to pull out a few of these families, we saw that cyanobacteria, which is this top row or bar here, methanotrophs, which are here, as well as human gut microbiota tend to be more abundant in the Elkhorn River. So again, very stark differences in this structural component. So now we're going to switch gears a little bit and talk about what's happening functionally within these rivers. So this is data that we are modeling what the diet contributions of different energy sources are within the river using these stable isotopes. And what you can see is that when we're looking at the invertebrates in these rivers and what they're eating, there isn't a whole lot of difference in how that energy is flowing between these two rivers. So regardless of whether regardless whether the invertebrates were pulled out of the Elkhorn River or the Niobrara River, we saw that they were eating basically mostly organic matter. Up to 80% of their diet was contributed by organic matter in both rivers. We also saw that the invertebrates are breaking down organic matter at similar rates in the two rivers. So these points on the left is for the Elkhorn, this is for the Niobrara. Briefly, we set out cottonwood leaves in both rivers in mesh bags, collected them over time, and then this is how much they lost mass over that time period, and then we calculated a rate from that. The two rates are shown here. They're pretty similar to each other, and there's strong overlap between them, so we don't really detect a difference between organic matter breakdown rates in these two rivers. But if we look at the function of ecosystem respiration and gross primary production, what I'm showing you here is what I like to call a fingerprint, or what's being called a fingerprint of these two variables that are happening through time in both rivers. And the Elkhorn is shown in these blue surfaces, and then the Niobrara is shown with the red surfaces, in which you can see that there's some pretty strong differences in that metabolic regime between the two rivers, where the Elkhorn tends to be way more productive and also respires a lot of carbon as well. And then there's more clustering of the data for the Niobrara, where they tend to be more equal in how gross primary production rates and ecosystem respiration rates are operating in these two rivers. So a stronger difference in ecosystem metabolism than we're seeing for the food web energy flow. So just take a step back here and kind of unpack some of this. What we're seeing, we think, and this is kind of our initial ideas, or at least my initial ideas, are that we have pretty distinct structural differences between these two rivers, especially in terms of water chemistry and the microbial community. So there's some really strong responses to what's happening in the landscape in terms of water quality in the microbial community. But the functional differences were more subtle, especially for the food web and organic matter breakdown. And we think that using these kinds of multi-scale integrated metrics is revealing things about key signals of human impacts in terms of organic pollution and eutrophication, but that the feedbacks to functional properties might be a little bit more muted, but they help us establish baselines. Since these things are happening through time, then we can figure out, okay, do we see a dip in one of these functions that might tell us that something is happening structurally that we might want to look at more closely. So thanks for listening, and please reach out if you have any questions. So thanks.
We have plenty of time for questions. I was wondering, did you measure biomass, standing stuff?
Of the basal resources, we did not, but I think that's on our to-do list for the
Okay.
next. Or we do have
We do
astery
have-
dry mass of biofilms.
Yeah, so we have biofilm, astery dry mass, and chlorophyll, and it's about 10 times higher in the elkhorn.
Okay.
than the Agri.
Was wondering.
Yeah, thanks. We don't have organic matter, which I meant to get. I think it'd be helpful.
How many data samples were collected? Can I know about that? Particular nitrogen?
So the question was how many data samples were collected? And then what was the second part of the question?
That was my question.
Okay, so kind of dependent on which level we were thinking about. So for graph samples of water, we basically had 4 sampling stations in each river. But then for some of the for the macroinvertebrates, for example, we went to each of the 4 stations, but then we collected multiple macroinvertebrates. So the sample size is varied there. And then for the sensor data, we have every 10 minutes. So it kind of varies depending on which level you're thinking about.